My name is Carolyn Coyle, and this is my presentation on macromolecules moving through the cell for Bio 1050. The movement of macromolecules. This video is about how steroid hormones, protein hormones, and intracellular proteins travel through the cell. Starting with steroid hormones. The steroid hormone that I am using as an example in this video is progesterone, although most steroids begin the same way with a precursor molecule of cholesterol that gets metabolized and undergoes alterations within the cell to become the functional hormone that it is meant to be. We will start, this flowchart is a little bit misleading, I will say. Number 1 should be in the nucleus in this purple circle in the center there, because as we know, everything starts with DNA. So the nucleus writes DNA, it is transcribed into RNA, which is then translated to mRNA to move out of the nucleus to the bound ribosomes on the endoplasmic reticulum, which is my 1 label there. Then from the endoplasmic reticulum, it is sent over to the Golgi apparatus, and the Golgi then sends it over to, in this case, the outer mitochondrial membrane. And there are certain proteins and enzymes involved in allowing passage from the outer mitochondrial membrane to the inner mitochondrial membrane, which I've labeled in a flowchart in the next slide here, but I'm not going to focus on that as much. Once inside the mitochondria on the inner membrane, the cholesterol is metabolized into a steroid called pregnenolone. And the pregnenolone is catalyzed into progesterone, my example here. It does need some help from the smooth endoplasmic reticulum, as I've labeled here. It kind of goes back and forth exchanging intracellular particles to get those catalyst activation things happening. Once it is fully turned into progesterone, then it goes to bind to the cell membrane, and it exits through the cell membrane because it is lipophilic, as all steroids are, and it easily passes through the plasma membrane out to the bloodstream, where it can travel through the body and do all of its steroid hormone jobs. Here is the written description flowchart corresponding to the numbers in the the illustrated flowchart in the previous slide. I have one of these written flowcharts for each image that corresponds to the numbers, although I'm not going to go through them in detail. The next macromolecule we'll talk about is protein hormones. The protein hormone that I am using as an example is insulin, which is created or synthesized in the beta cells of the pancreas. As we can see in this illustration, the cell's nucleus writes the DNA, and that DNA is transcribed to RNA, which is then processed to become mRNA that can be transported out of the nucleus. The mRNA travels to ribosomes on the rough endoplasmic reticulum, where it is processed and sent over to the Golgi apparatus. The Golgi packages all of that good information into secretory proteins, which are then shipped out of the Golgi apparatus towards the membrane, the cell membrane, and it kind of binds to the cell membrane, where it is able to then secrete its materials, in this case insulin, out of the cell. Here is the written description flowchart of that description that I just mentioned in the previous slide. Lastly, we have intracellular proteins. Intracellular proteins are exactly what they sound like. They do all of their work from inside the cell. These proteins do not ever leave the cell, and they also do not utilize the endomembrane system in the same way that the other macromolecules that I previously discussed do. As an example, I am using actin filaments or actin-binding proteins, globular actin that becomes actin filaments that become actin cytoskeleton in the cell. So to start again, the nucleus writes DNA for this protein. It is written into RNA, then mRNA, and then differently from before, the mRNA information is then sent over to free ribosomes in the cytoplasm rather than the endoplasmic reticulum. So from these free ribosomes, it is able, or within these free ribosomes, the mRNA information is synthesized into proteins, in this case actin proteins, globular actin to be exact, which can bind to itself. Multiple G-actins kind of become this one elongating actin, and it eventually becomes an actin filament after enough is added to each end. And so those actin filaments can then cross over each other and form a kind of layer inside of the cell membrane called the actin cytoskeleton. And this just serves to add support, shape, structure to the cell. These proteins do not ever leave the cell. They just serve as, intracellular proteins. They stay inside the cell and do their jobs from within. And finally, we have one more written description in a flowchart form corresponding to the last slide. And these are my references. Thank you so much for listening.